थोड़ी बस मस्त आहे पण नाही Krishnanjali, you can start.
Okay, sir. Okay, sir. Okay. Sir, I'm audible, no? Hello, sir. Hello.
Yes.
Okay, sir. Good afternoon to one and all. Dr. A.M. Deshmukh, sir, President of Microbiologist Society, India. Today's webinar speaker, Dr. P. Maheshwari ma'am, and all other participants attending this webinar, I welcome you on behalf of Microbiologist Society, India. Today, we are all gathered here for attending PhD webinar series. Till now, we have completed 23 lectures. Today is 24th one. And speaker for today's webinar is speaker number 24, Dr. P. Maheshwari ma'am, Assistant Professor, Shri Sai Ram Engineering College, Tamil Nadu. Her topic for a webinar is synthesis, characterization, and antibacterial activities of pure and biomodified titanium dioxide nanoparticles. So before we start webinar, I would like to give short introduction of Dr. P. Maheshwari ma'am. Dr. P. Maheshwari ma'am, currently she is working as assistant professor in Department of Physics at Shri Sai Ram Engineering College, Chennai. She had received gold medal from University of Madras for her academic excellence in her for her undergraduate program. Also, she stood first rank in her postgraduate degree program. She had completed her PhD in the field of nanotechnology, and her research interests include synthesis of nanomaterials, solar cell, drug delivery, antibacterial and anti-cancer studies. She had published several papers in highly reputed journals, received many awards in her research career. She has attended more than 400 seminars, conferences, workshops from all over India. I would like to quote one thing: as universe arise questions, it gives answers also, which are next to us. I hope you will get the answers of all the questions which are in your mind, and I hope that you enjoy this lecture. So now I request Dr. P. Maheshwari ma'am to continue or start her webinar speech for today. Okay, ma'am, please continue.
Yeah, thank you, ma'am.
Welcome.
Thank you for the wonderful introduction you have given to me.
Thank you, ma'am.
Yeah. Can I start my presentation?
Yes, ma'am. Sure, ma'am.
Good afternoon to everyone present here. My topic regarding about this webinar will be synthesis, characterization, and antibacterial activities of pure and biomodified titanium dioxide nanoparticles. Actually, when we were undergoing our PhD, my thesis comprises about antibacterial and anti-cancer activities. So here, based on the time limit, I have made my part only with respect to antibacterial activities. My overview of the presentation, it includes about the introduction part, the synthesis methodology, the characterization, the application based on this, and the summary of the presentation. When we talk about introduction, now let me show you a small picture about this. We are all living in Earth. I hope that everyone. There is some kind of climatic changes, pollution, And there are several factors which are affecting the earth. And we can see that it is heating up. Sometimes it is melting. There is a nature of coughing. So many kinds of wounds. Several diseases are occurring. These diseases, when you talk about, like diarrhea, vomiting, typhoid, so many nerval disorders, skin diseases, all these things we are seeing here. And obviously, now what is the life of the earth right now? We need to save the earth, the life, the living beings, the human beings here. Now a question is posed to us, whether we want to live in this condition or we want to enjoy a life which is having like this. Not only myself, every one of us will say, we want to live a happy and a peaceful life. But how this is possible? Because every time you see, right now also there are several diseases everywhere occurring. It's because of water, it's because of air pollution, noise pollution, several pollution is happening over here. We are also taking so many controls, the government is also taking so many controls over that. But obviously the diseases are also there. And when we talk about the diseases, particularly in our part of webinar, we take around the diseases that are naturally the main sources for them, bacteria, viruses. We can't totally eliminate them. We are finding out so many kinds of medications, so many medicines are available. The objective of this particular part, all these medicines which are having right now, it is purely curing us? Yes, it's curing us, but at the same time, there are several side effects which is happening over. And all these medicines are not readily available. It's not abundantly present. The cost of that is very, very, very high. When you talk about cancer and all, the medications for cancer and all, it's nearly very costly. Can a poor guy imagine to buy such a kind of costly things? So in our part of this PhD purpose, we try to find out the drug which can be somewhat cheaper in cost level. And also, why can't we use the sources that are available on the earth surface, which is readily available with a cheaper cost? So that was one of the main objective which we have taken across. Okay, for finding all these things, we should have some kind of technology. What is the technology that is available? Now here comes the beautiful part, the emerging, the total world is emerging around this, which is nanotechnology. A small size, making the whole property to change over the whole world. And we are making use of this nano. Now, what does actually mean by nanotechnology? It's a kind of applied science where the whole thing we are making as an atomic or a molecular scale. So based on that, we put a field there that it comes with a technological evaluation. So when we talk about this nanotechnology, there should be some kind of synthesis proceedings. What are those things? When we talk about the synthesis, we have two kinds of part. One we call it as top-down approach, and another one we call it as bottom-up approach. Bottom-up means the small atoms, they try to combine together to form clusters, and that it tries to form as a nanoparticles. Whereas in the case of top-down, a bulk material we will be taking it across, and that has been made as a powder. From the powder, you try to get the nanoparticles. Actually, these are the two extreme levels of synthesis technologies that is available in the case of nano. Now, when we talk about the top-down, there are some process called ball milling. All these things can be happening. In the case of bottom-up, we use chemical reactions, particularly sol-gel synthesis, hydrothermal synthesis, spray analysis, so like that, pyrolysis, we will be taking. Now, in the case of our particular preparation, we prefer to bottom-up technology. So under that bottom-up, we preferred one synthesis, which it is named as hydrothermal. Now, why we have chosen this methodology? This method it will make, it is a chemical, it's a heterogeneous reaction where the precursor or the starting material which we are using, it is inexpensive. It's not high costly. At the same time, you can control the size and shape of the particles which you are going to prepare. So these are the two things why we prefer the hydrothermal methodology. Now we require some kind of instruments to undergo this hydrothermal technology. So this is the one particular part. You can see this is we normally call it as a Teflon lined autoclave. And these are the internal parts which we have. And you can see the precursor is loaded here. You have a stainless steel. And apart from that, you have a Teflon lining. This Teflon lining is actually a poly carbon. Now you have to make this in particular part. You can heat the temperature around nearly about 200 degrees centigrade in this, not more than that. Okay. You can maintain that particular temperature, and whatever you are going to have, you can control the whole morphological parameters by using this. And you will be having a lid over this, which after making this liquid part, you just keep it in your furnace, and then you can take part with all the synthesis technology. So this is the methodology which we preferred around for our part. So the precursor which we are using is totally inexpensive. And normally it will be taking place in the presence of a solvent. You can either use an aqueous solvent or a non-aqueous solvent. Okay, now I have made out, this is a synthesis technology. So my part is, I'm going to take about nano. In the nano, I have made my mind, okay, let me start with the hydrothermal technology. Now in the hydrothermal technology, what is a particle? Which one I'm going to have it? So my application is something with respect to bio-related. So I need to have it as a biocompatibility. It should be. So under that condition, when we undergo for a literature survey, we found that titanium dioxide nanoparticles, they have been proven to be an excellent photocatalytic material. It's a best material in the case of photocatalysis. So, and we thought that why can't we use this particular? And also, it is somewhat biocompatibility in nature also, because in so many dental applications, they are trying to use this particular part. So that is the reason we have chosen this. These are the properties of titanium dioxide nanoparticles. And apart from that, the powder looks like this. These are the properties why we normally choose for our application part. Because of their high surface area, their small crystalline size. Because when you have the crystalline size to be very small, it can easily penetrate into any of the tissue layers. And also the anatase form of titanium dioxide is a very good photocatalytic part of it. The high crystallinity structure and the porous nature, and also the activation of the light source. So these are the main important characteristics why we prefer this particular material for our process. Now, once we made out all these things, then we have to go for experimental technology, because that is the evidence which we have a proof. So once we make out the particle, we have to understand whether it is a nano in range. Okay, when it is a nano, what kind of structure it is having, what kind of parameters it is having, and how the absorbance nature is, what all the functional groups it may be present inside. So we have to need a part of it. And we also should understand about what is the size of the particle we are getting. So we undergo these experimental technologies. The X-ray diffraction method. Actually, we call it as XRD. Now, this particular methodology is very, very helpful in the case of how to find whether a material is crystalline or not. And the next part is UV analysis. Using the ultraviolet rays, we were able to find out what is the maximum absorbance a material can withstand. And also from the FTR analysis, we can determine what all the functional groups that are possibly present over here. And also the TEM. TEM is a transmission electron microscope where it's full of electrons. Okay, the electrons are the sources here, which they will be determining the morphological structures of this analysis. And it is one of the best methods to determine the particle size of this nanos. And then based on all this experimental, the next thing is our application part. And here we are moving towards bacterial. How to somehow inhibit the growth of bacteria or to kill that bacteria. So that kind of activity, we call it to be as antibacterial activity. So here we will be using the agents, antibacterial agents, which the drugs we are finding out. And based on the drugs, we just check out whether it is bactericidal, whether it kills a whole bacteria or it will inhibit the growth of the bacteria. So basically, when we talk about antibacterial nature, we should be aware of what is a bacterial bacteria and what is the type of bacteria. So based on that, we have generally two categories. One, we categorize them as gram-positive, and the another one, we categorize them as gram-negative. In the case of gram-positive, you have a thick, there's a brown color region here, which is a thick peptidoglycan layer. Whereas in the case of gram-negative, that thick layer which you saw here, it will be very thinner. And there is one more membrane which is present in the case of gram-negative. We call it as an outer membrane. This outer membrane is fully comprising of lipopolysaccharides and proteins. Now, what our drugs has to do is they have to enter into this membrane inside. They have to try to stop the synthesis, the food which they are giving, that they have to stop it. So that will end up the DNA of that material and it tries to kill the bacteria. So this is what is the mechanism behind that, which we are using out. So in our experimental part, we used 3 gram-negative bacteria and 2 gram-positive bacteria. These are our test organisms which we are using around: Escherichia coli, Pseudomonas Klebsiella pneumonia, and Pseudomonas aeruginosa. These are the 3 gram-negative bacteria. And Staphylococcus aureus and Streptococcus mutans, this comes under the category of gram-positive. So in our part of the thing which we have done experimentally, we used these 3 species and these 2 species in order to make out a. Now there are several tests for this antimicrobial part, and among that we have chosen this disc diffusion technology, where obviously the bacterial culture will be there and it will be in a petri dish. We take the agents in the form of a disc shape and we place them. We incubate it nearly for about 24 to 48 hours so that you can see the zone will be formed over. That zone of inhibition, we need to calculate it, which clearly tells us how much amount the growth of the bacteria has been inhibited. So based on that, we have taken the titanium dioxide and we made the synthesis technology. Now let us go for the synthesis which we have done in our experimental part. So this is a synthesis technology which we have done it. The precursor, the main material for this, we have chosen titanium tetraisopropoxide, and then along with that deionized water and isopropanol. We were mixing it for nearly about 2 hours in a magnetic stirrer, and then the contents were transferred into the autoclave and it was maintained at 200 degrees centigrade. And after that, we have centrifuged it and dried it with oven, and the particular powder was annealed around at 350 degrees centigrade. Because our target is we need to get the anatase form of titanium dioxide. So we were annealing it around 350 degrees centigrade. And after that, the particles which we have obtained, it is titanium dioxide. Now this is experimentally we have done it. So we need to do some experimental for characterization. Okay, this is a confirmation. This is what I have to do. So for that, we went for the XRD diffraction, where we got this particular data. From the data, the plot has been assigned here. Now we can see clearly, based on the XRD pattern, there are sharp peaks here. Now these sharp peaks clearly indicate that they are highly crystalline in nature. And among these planes, we can see 101 plane. This is obviously coming under anatase. And there is a thing called JCPDS, which is Joint Committee on Powder Diffraction Standards. All the data which have been a standardized part, it will be put in this bank. So from that JCPDS file number 211272, it exactly matches with this pattern. And also we have a mixed phase here. A small fraction of bruchite is also available. This again, it is matching with the JCPDS file number of 761936. So from the XRD pattern, we have a clear thing that they are highly crystalline in nature. Then we went for the UV analysis. From the UV analysis, we were able to see the maximum absorbance of our synthesized particle. It was around 264 nanometers. Now, in the case of FTR spectra, from the data that is available here, we can see this is normally known as a fingerprint region of the IR spectroscopy. So under that condition, you can see 633. This forms a very important, it's a strong stretching vibration of the TiO bonds. So based on this. And apart from this, this is a TEM and HR-TEM analysis. This is a most highly preferable one and a highly confirmation part of it. So in the case of TEM analysis, you can see in 100 nanometer resolution, this is a one we can see around the picturized part of it. And HR means it is a high resolution where you can even go for 5 nanometers and all. So under that, now you can see there are patterns. These are the diffraction fringes, the lattice spacing we call it as. We can see this clearly here. So when we took the measurement of this, we found out the spacing comes around as 0.352 nanometer. When we were manually working with XRD, this really matches clearly. And from that, the particle size was estimated to be as 7.5 nanometers. So this comes under about our synthesis of TiO2 nanoparticles. Now, comes to the antibacterial. Now, in the category, we have found the zone of inhibition for our synthesized nanoparticle. The gram-positive Streptococcus mutants showed the highest zone of inhibition, where nearly about 10 millimeters.
Hello? Hello? तुम पार्सल घेऊन आला ना इकडे माझी मीटिंग चालू आहे.
Can you please mute yourself?
Hello?
Yeah. In the case of the pure titanium nanoparticles, ma'am, can you please check out who is talking there? Kushanjali ma'am? Kushanjali ma'am? Yeah. So when we talk about the zone of inhibition here, we can see that the Streptococcus mutants, they have given nearly about 10 millimeter of zone of inhibition. So now there is another question for us. Why can't we make some more modification for this titanium so that it can exhibit more excellent antibacterial nature? Now we were searching for the sources and we have found that the aqua rosa and the protein powder, why can't we modify it along with the titanium dioxide nanoparticles? So this was the next particular part which we are going to make it. So again, we went for the synthesis technology where the titanium tetraisopropoxide along with isopropanol and deionized water, it has been transferred, it was mixed together for nearly about 2 hours. And then we have taken to the autoclave at 200 degrees centigrade. And then after centrifuging process and drying it in oven, then we have annealed it around 350 degrees centigrade. So we got the nano powder. Now we have taken the aqua rosa. It's a normal, we call it as paneer roses, okay? So that aqua rosa, we have taken it and we have made them as an extract. Now that extract, it was mixed with this nano powder and it was using a magnetic stirrer. We have stirred for several hours and then we centrifuged them, we dried them, and then we made it as a powder. That powder comes under the category of aqua rosa modified titanium dioxide nanopowder. Similarly, we took protein powder extract also and mix it with titanium dioxide nanopowder, and we yield the result to be as protein powder modified titanium dioxide nanopowders. Now again, we have to check whether it is how the pattern is, XRD pattern. So when we have out the XRD for aqua rosa and for protein powder, we have seen these particular things. You can see the intensity here, where the 101 plane, this is for our synthesized titanium dioxide nanoparticles, and this is the aqua rosa modified one. And this will be the protein powder modified. So when we see this, there is no change in the XRD pattern. There is no peak shift which we have observed here, because whatever the material which we have added here, they are not acting as, they are not changing the total lattice. They are just modifying only the surface. That is the reason you can change the, you can see, you can observe the intensity alone is decreasing with respect to addition of modifiers, and there is no peak shift that is obtained over here. Now, in the case of UV spectra, we were able to see that in the case of pure titanium, which we have synthesized, the maximum absorbance is 264 nanometer. Whereas in the case of protein powder and aqua rosa, because of the modification, we were able to see there is a decrease in the maximum absorbance. And also we can see that there is a shift here to the blue, which means we call this to be as a blue shift. This blue shift happens because of quantum confinement, where the size of the particle becomes very smaller. And this we can clearly confirm that with the TEM images which we have obtained for this. Now, in the case of FTR spectra, in aqua rosa, the main component which is responsible for these antibacterial nature, the compound is known as geraniol. There are several compounds which is involved in the aqua rosa extracts. Among that, the geraniol has been proved to be a good antibacterial nature, which is enhancing the nature in that aqua rosa. So this geraniol compound, and this is a spectra. This is the spectra for the aqua rosa extract alone. And this is for the protein powder extract alone, the FTR spectrum. So you can see here where we have 3416, we can see the OH vibration of geraniol compound. And in 1444, we see C double bond C vibration of geraniol. And similarly, the CO vibration of geraniol is found around at 1130. So here we have made out where geraniol compound is. And similarly, in the case of protein powder, it is a combination of amides and amines. So we can see here 1629, it has a CO vibrations of amides, and in 1390 we have the CN vibrations of amides, and also in 1057 we have the CO vibration stretching of amines. Now these are going to play a major role when it is modified with TiO2 nanoparticles. So this is the one which we have seen here, and you can clearly see from this. Now I have taken another picture for us to show this. So this is the one. So now you can see from these two parts, the red color line is a synthesized TiO2, and the green color will indicate it is an aqua rosa modified, and the blue color gives you it is a protein powder modified. You can see more or less the aqua rosa and the TiO2, they are in all the level, but you can see there is a difference in the fingerprint region, and also you can see there is a change in this particular. There is a broad peak. There is a peak over here, and also the peak is becoming like this here, which indicates the compound has really acted with TiO2. And also when you talk about the, you can see a drastic thing here. And also around 1634, you can also see there is a shifting. there is a broader peak around 1393 and 1068. Now, which means the amide and the amine group of the protein powder, they have reacted with TiO2. Now, from the TEM and HR-TEM analysis, now we can see for this particular part. This is the normal TEM analysis, and this is a high resolution TEM analysis for the synthesized TiO2. And this one, the Aquarosa modified TiO2 for TEM, and this is for the HR-TEM. And similarly, when you talk about here, this is for protein powder modified one, and this will be the HR. That is, when you want to go very closer, a very high resolution, you go to HR-TEM analysis, where you read around in 5 nanometer scale. So in all the things, you can see there is a spherical shaped TiO2, and you can see when the modifiers are added over, there is a high level of agglomeration, which you can see over here. And the particle size have been estimated to be as 7.5 for the synthesized one. And for protein powder, it is protein powder modified, it is 6.5. And for Aquarosa, it is 5.5 nanometer, the particle size. Now coming to antibacterial nature. Now, in the case of antibacterial, we can see clearly there is a very good enhancement in the antibacterial activity of the protein powder. This is actually protein powder. The control which we have used here is streptomycin, okay? And when compared to the control, we see that the gram-negative bacteria, they exhibit more zone of inhibition when compared with the gram-positive. This is because in the protein powder, you have the amides and the amines, and they are nothing but proteins. They are the lipopolysaccharides. So they were able to easily penetrate into the outer membrane of the gram-negative. So that is the reason more number of particles and more number of particles were able to enter into the more number of bacterial cells. Now, in the case of Aquarosa, we can see the zone of inhibition was able to see only in the gram-positive, not in the gram-negative. So the geranyl compound was able to enhance, but it was able to enhance nearly about 11 millimeter. There was a slight change in the case of this, but moreover, when we talk about the addition of the modifiers with the titanium dioxide nanoparticle, it has enhanced the antibacterial activity, which means it was able to inhibit the growth of bacteria. More number of bacteria were able to kill by making the addition of these modifiers. So actually what I have told you about all these things, it is only one part of my chapter, which I have did in my thing. I have done it nearly about 7 chapters, where several sources I have taken, and we have also used for anti-cancer activities also, which it played a major role. And we found out nearly about 97% it was able to kill the cancerous cells. And from the summary, I can conclude that the synthesis have been done for pure Aquarosa and protein powder modified titanium dioxide nanoparticles. And the powdered samples, they were undergone or subjected for characterization using XRD, UV, FTIR, and TEM analysis. And the XRD spectroid shows that the pure and the modified, they are highly crystalline in nature. And in the case of UV spectra, we can see the absorbance has been decreased with the addition of the modifiers. And on the whole, the conclusion is the protein powder which was modified along with titanium dioxide nanoparticle, they were acting as an excellent antibacterial agent when compared with the pure titanium dioxide nanoparticles. And these are my list of publications. And these are my list of conferences which I have attempted. And these are my references. Any questions, please?
Ma'am, have you done your lecture? Should
Yeah,
we start
ma'am.
for question?
Yeah, yes, ma'am.
Okay. Thank you, ma'am, for giving such good lecture. I think she has explained all the points. And I think now we should go for a question answer session. If is there any question you may ask?
Yeah, sure, ma'am. Any questions, please?
Ma'am, your lecture was excellent,
Thank you, sir.
and it is now recorded on the YouTube,
Yes, sir.
and you will receive certificate and one memento
Yes, sir.
on your address, home address.
Yes, sir. Sure.
So you send your home address to me,
Okay, sir.
and we'll send the certificate to you. And the most important thing is that those who want to do PhD in similar topic or your topic, definitely they will refer your YouTube in the future.
Yes, sir. It's always a welcome thing from such a great people like you. I also need such kind of collaboration. I always welcome that.
Sure, sure. Sky is the limit in our subject.
Yes,
Correct?
sir. Yes, sir.
Yes, yes. And we'll do maximum things in the subject even in the future.
Yes, sir.
You are in Tamil Nadu.
Yes, sir. I'm from Tamil Nadu in Chennai. Actually, sir, in my topic, it is full of with antibacterial as well as anti-cancer. Now, since the time limit was only for 30 minutes or 1 hour, so I just made my part to be only for antibacterial. And also another thing is, we did for several material like this. I have just given only for aqua rosa and protein powder. Like this, we have did nearly about some 20 to 25 sources. And all these things which I could not explain it here. So I just want that whatever we are giving out, these inputs will be helpful for others also. So they try to get benefit from us, and they try to develop any technological, which it will be in turn useful to our society. That is the first thing which I want to make out here. That is it.
Very good. Very good. Krishnanjali, there is a one question in the chat box.
Yes, 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 sir. Ma'am, there is one
Yeah.
question. Which protein powder you have used? The question is from Rakshanda Singh.
Actually, sir, the protein powder which was available in Tamil Nadu from Chennai, we have taken that powder, okay, and it was one of the costliest powders since it is what I can say to you. Since it's a YouTube live, I cannot name the company, but it was one of the protein powder that is made available in Chennai, and it is highly costly also, and it is given for babies. So based on that, we have taken that survey and the results. And I can give you one thing. It comprises of lysine. Okay, there are nearly about 6 amino acids involved inside.
Okay. Is there any question from other participants?
I think there are no questions. You can conclude now.
Sir, just a minute. Can I give some acknowledgement also?
Yes, why not?
Yes. First and foremost, I thank my supervisor, my guide, Dr. S. Ponusami sir, Professor of Physics Department, SRM Institute of Technology, Katangalatu, Tamil Nadu. And also, I convey my thanks to Professor Dr. Hayakawa sir, Japan, Shizuoka University, for giving us so many facilities to undergo this research part. And I convey my thanks to my college, Sri Sairam Engineering College, Tamil Nadu, for contributing my research work and also to give a webinar like this. And the last but not the least, I have to convey my sincere thanks to the Microbiology Society for giving me a golden opportunity to convey my research work in my field to deliver this webinar. And my special thanks to Jai sir and to Deshmukh sir and also Prashanjali ma'am. Thank you. Thank you for everyone present. Thank you, sir. Thank you.
Okay. Now it's time to thank you, everyone. First of all, I thanks to our today's speaker, Dr. P. Maheshwari ma'am, for giving such a beautiful lecture. And also thank you to Dr. M. Deshmukh sir and all other participants. I hope you enjoy this lecture. Thank you.
Yeah, thank you, Rakshanda Singh, sir. Yeah, thank you. Thank you, ma'am. Thank you, sir.
Thank you, ma'am. Sir, should we end the meeting? Hello, sir.
Ma'am, I think sir's bandwidth is very low.
Okay, ma'am. Then it's. I think we should end this meeting.
Yeah.
Okay, thank you, ma'am.
Please, yeah, thank you, ma'am, and please convey my thanks to Jai sir also.
Okay, definitely.
And in future, if there is anyone who is interested in working in my area, I just give my mail ID also. They can contact me. Just a moment, ma'am. I'm typing my mail ID.
Okay, ma'am.
Okay, they can contact me.
Okay, ma'am. So ma'am has given her email ID. Anybody interested in this topic in future to work, you may contact her. And I think we should now end this meeting. Is there any question about this? She has given her email ID. I think no problem. Ma'am,
yes,
you leave the meeting. Okay.
Yeah, thank you, ma'am. Thank you all.
Okay.